everyone, I am Marilisa Benedicto, and today I will be explaining Benedict's test, a simple yet important biochemical test used to detect the presence of reducing sugars in a solution. Although I won't be performing my experiment by myself, but I will walk you through the process and explain the results in detail. The main objective of this test is to determine whether a carbohydrate solution contains reducing sugars. We will identify the sugar based on the color changes observed after adding Benedict's reagent and heating the solution. So, to perform this test, we need 7 test tubes containing different carbohydrate solutions, Benedict reagent, and the boiling water bath. The main ingredient here is a Benedict's reagent, which reacts with the reducing sugars to produce a color change. Benedict's test is a chemical experiment used to check for the presence of reducing sugars. Reducing sugars are the types of carbohydrates that can donate electrons in a reaction. Now, let's go through the procedure that step by step can explain what the Benedicts are. First, we label our test tubes based on the carbohydrate solution we are testing. This is the most important so we can identify which solution is in which the test tube. The second one, we use a dropper or pipette to add a 1 milliliter of each carbohydrate solutions into the separate test tubes. So this ensures that the amount of liquid is in the same in all test tubes so the test is accurate. The third, we add a 1 milliliter of Benedict's reagent into each test tube. Benedict's reagent is a bright blue liquid because it contains copper sulfate, which has a copper, sulfur, and oxygen. The fourth, after that, we gently mix the solution to make sure that Benedict's reagent spreads evenly in the test tube. Now, the fifth is we place the test tubes in the boiling water of 100 degrees Celsius for about 2 to 3 minutes. This step is very important because heat makes the reaction happen between the Benedict's reagent and any reducing sugars present. The sixth, after heating, we take out the test tubes and let them cool. Then we carefully check if the color of the solution has changed. The color change will tell us if there are reducing sugars and how much is present. So let's take a look at the result for glucose, fructose, and xylose. Glucose, we observe a positive result for both color production and precipitate formation. This indicates a moderate reaction. Similarly to fructose, also shows a positive result for color and precipitate formation, leading to a moderate interpretation. And the xylose stands out with a positive result of color production and a higher amount of precipitate formation. It's indicating a strong and high interpretation. So now let's review the result of lactose, maltose, sucrose, and starch. For lactose, we see a positive color change and precipitate formation indicating a high reaction. Maltose also shows similar results with a high reaction interpretation. And the sucrose produces a color but no precipitate, meaning there is no significant reaction. Likewise, starch shows a color change but no precipitate formation, resulting in no reaction interpretation. So the Benedict's reagent has a chemical called copper sulfate. When we heat it with a sugar that can be reduced, a reaction happens. The copper changes from one form to another, creating a solid called copper oxide. This solid is what gives us a color change that tells us that if sugar is present. In summary, Benedict's test helps us to identify reducing sugar based on the simple color change. This test is widely used in medicine, food, science, and the biochemistry. Thank you for watching.